It's my pleasure today to be the host of the Diversity in Biology seminar series. I'm Julien Gibon, assistant professor in the biology department at UBC Okanagan. We would like to start by acknowledging the ancestral territories on those on land we are currently living and working. We anticipate that since the event is in UBC Okanagan, many of attendants are in Kelowna, on the ancestral and unceded territories of the Syilx Okanagan Nation. Many of you are joining from other regions, including our speakers. So we encourage you to reflect on the land and on which you live and work, and who the ancestral caretakers of those lands might be. So today it's my privilege now to introduce our speaker, Dr. Leo Ng. Leo was raised in Richmond and did both his bachelor's and PhD at the University of British Columbia. He was trained under Dr. Eric Asseli, and having biochemistry background, he took on the project that involved the cyclic nucleotide binding site of the pacemaker channel. He then did a postdoc at Northwestern University under the mentorship of Dr. Paul Dequin, a renowned expert in electrophysiology. I think you learned to do a patch clamping there, and now it's a technique you use a lot. So today he will share on one of those projects, which show the structure and blocking mechanism of tamoxifen on voltage-gated sodium channels. So before we listen to this fascinating talk, I have a few things to share. If you have questions, you can post your question on the Q&A at any time, and there will also be an option to raise your hand at the end of the talk and have the mic on if you want to ask your question. I would like to remind everybody that the event is being recorded, so audience members that do not wish to have their voices recorded should opt for the Q&A and typing questions. Without further ado, I will give the floor to Dr. Leo Ng for his talk about tamoxifen effect on voltage-gated sodium channels. Dr. Leo.
Yep. So let me just share. Can you all see the PowerPoint? So let me do full screen.
Very well.
Okay. So first, so good morning. And first, I'd like to thank the organizing committee for this kind invitation. Thank you, Julian, for the introduction, and also for everyone who's listening today. So as said, I graduated from UBC a few years ago. And so it's my great honor to be able to, I guess, come back to my home institution and be able to share my research experience. And hopefully this might be like an inspiration for some of the, I guess, younger scientists, or I guess our next generation scientists. So I initially thought that the word diversity was referring to the ethnicity of the speakers, but I also found it very fitting to the wide spectrum of how we have a wide spectrum of topics here. And I'm guessing that I'm going to be representing the physiology/pharmacology group. So in the physiology lab, typically we identify a protein of interest that we want to work on, and then we will just hammer on it and just study it, characterize it, learn everything there is to learn about that protein. And in order to get fundings, there's always a need to write the significance and also somehow link it to a human disease, and then maybe study the human disease mutations associated with those and how the disease forms because of mutations in this protein. And a step further might be then to find different therapeutics that might regulate this protein and possibly prevent the progression of this disease. So in my PhD at GBC, under the guidance of Dr. Eric Asseli, I worked on the pacemaker channel. So his role is to generate and to regulate the heart rate. So it's no surprise that the associated disease would be cardiac arrhythmia. And then later on, or cardiac arrhythmia, which is the heart beating at an irregular rhythm. So then later on, I went to do my postdoc at Northwestern University under the mentorship of Dr. Paul Dequin. And there I did two different projects. So one of them was on the protein found in the kidneys, and it's mechanosensitive, so then it detects the fluid flow in the collecting ducts of the kidney. And the associated disease is autosomal dominant polycystic kidney disease, or short ADPKD. This picture might be a little disturbing, so you might want to turn away if it makes you sick. So this is what ADPKD is. So on the left is a normal kidney, and on the right is a polycystic kidney. So someone with this disease would have these cysts formed in the kidneys, which is impeding their normal kidney functions. And then the final proteins that I worked on in Paul Khan's lab is a protein in the nervous system, and it's responsible for transmitting signals from one place to the other. So for example, maybe it's transmitting signals from the brain to my hands right now. And disorders with that protein could be linked to epilepsy or chronic pain. So at this point, you might be wondering, what does all this have in common? So I'm going to mainly focus on this third protein today, and also slightly talk about my PhD project as well. And the link between all these three projects or proteins is that all of them are ion channels. And so you probably all know that a cell is encased by the phospholipid bilayer, and it's sort of like a barrier around the cell. So in order for ions to get in and out of the cell, it requires ion channels, or these integral proteins that allows proteins to go in and out like a door. And there are definitely many ion channels out there, and I've only worked on a handful of them. So they're different because they might have different ion specificity. They might be in different locations. They probably have different functions, and also different regulation mechanism. So the one that I'm going to focus on today first is called the voltage-gated sodium ion channels. And so it's a sodium channels, and therefore it's permeable primarily to sodium ions. And it's voltage-gated, meaning that the opening and the closing of the channel is regulated by voltage, or by the change in the membrane potential, or simply just the charge difference between the outside and the inside of the cell. So voltage-gated ion channels plays a fundamental role in the generation and the propagation of electrical signal for communicating in a neuron. And the process is called action potential. So here in the box, showing you a typical action potential, where the y-axis is basically the change in the membrane potential. So this action potential, I also have alongside with the activity of the sodium current. So you can kind of see at the same time what sodium channel is doing as the action potential is propagating. And at the time point right here at the beginning, when action potential is really just at rest, the sodium channels are also not doing anything. And at some point, there will be a stimulus. The stimulus could be chemical, could be mechanical, or it could be electrical. And that stimulus causes a change in the membrane potential, causes sodium channels to go from the open to the, from the closed state to the open state. And it also leads to an influx of sodium ions. So sodium are going to go from the outside to the inside of the cell. And because of that change in the ion, it causes depolarization of the membrane, which means that there's more positive ions on the inside of the cell, leading to this upstroke of action potential. So you can kind of imagine like a door, like an automatic sliding door, probably. So when the sensor senses something, it opens, and that's what's happening. And then afterwards, the repolarization, or the falling phase of the action potential, is partially triggered by the inactivation of the sodium channels. So you can see that inactivation is just a process when the channel on its own is going to go into a non-conducting state. And it leaves the channel in this refractory period. And again, with the sliding door, you might think that the sliding door is closing and there's a lag time before it can open up again, and that's inactivation. When there's an aberrant mutation that affects inactivation, it could like delay inactivation. You can imagine that there will be too much sodium channel activity now. And because of this extra channel activity, it will lead to extra action potential firing, which causes conditions like epilepsy or neuropathic pain. So it's not surprising that these channels are appealing therapeutic targets for anticonvulsants or like local anesthetics. And of course, what we hope to do here is to find a drug that can attenuate the issue with the inactivation, and then return the action potential back to normal. So pharmacology of sodium channels have been pretty well studied in both the human and the sodium version. So the human and the bacterial version of sodium channels. So the human versions of the channels are these gigantic proteins that consist of over 2,000 amino acids residues, and it spans across 4 homologous but non-identical domains, and they assemble together like this. So the bacterial version, in contrast, are way smaller polypeptides, usually around just 200 to 300 amino acids long, and that stretch just codes for one of those domains. But then those 4 domains, so the domains can also come together, assemble together into one tetrameric functional channel like this. So the advantage of studying the bacterial version is that the smaller protein is just easier to express, and also they would be easier to crystallize if we want to look at it structurally. The bacterial and the human versions are also quite similar in terms of topology, or in terms of how they look. So there are 2 important regions, and the first one is called the VSD. So among the 6 transmembrane domains, the VSD is the S1 to S4 of the transmembrane segments. And most importantly, I guess, is the S4, because it has some positively charged residues. And what it does is it senses the differences in the membrane potential, or changes in the membrane potentials. And it would actually physically move up or down to indicate the activation or the inactivation, activation or deactivation of the channel. So it requires the S4 movement, which then leads to a opening in the S5 to S6, which is the other domain, which is the pore domain, and it's the inside or the central cavity of the ion channel. So that's where the ion conducting pathway is. There's also a loop region between the S5 and S6, and we call that the selectivity filter, because that dictates what ions get to go through. Now, it has been shown that ligand binding sites between these 2 versions are also quite similar. So what kind of ligands or what kind of drugs would bind to sodium channels? So we know that spider toxin or scorpion toxins, they bind to the extracellular loop of the voltage sensor. Another one is tetrodotoxin or pufferfish toxin. So those bind to the selectivity filter right in the middle. So therefore, they basically just block the ion conducting pathway. Many local anesthetic, like lidocaine, they bind to a transmembrane fenestration. So that's a small opening between the membrane and the central cavity. And you can kind of imagine a hydrophobic small molecule like lidocaine can kind of just sneak through the membrane and goes into the binding site. So those are the known binding sites. And recently, well, I guess not recently, that's like 2 years ago now, our collaborator in structural biology, we were looking at the novel pharmacology of sodium channels. And we were looking at tamoxifen, because tamoxifen is found to have side effects to sodium channels. So after crystallization, we found that tamoxifen is at a novel binding site, different from all the other ones. And we also found that very unique biophysical properties of this drug that I'll go over in a bit. All right, so now we see that all these ligands bind to different sites. So we would imagine binding different sites probably have different effects on the channel as well. So the scorpion toxin here again binds to the voltage sensor domain. So it's actually going to alter the movement of the S4. And because of that, it would change the voltage dependence of activation. So here, what's happening is, in this case, the shift is going to make the channels easier to open. So that means there's going to be extra sodium current, which would enhance the sensitivity, the sensation of pain to the target. So those are what the toxins are doing. Now, there is also this Japanese pufferfish toxin that you should probably avoid eating. So those ones, like I said, they are physically blocking the ion conducting pathway because they block the selectivity filter. So because of that, we have a reduction in the ion conductance. So there's just less sodium current. And what that does is in the muscles, when that happens, you're going to have paralysis happening. So both of these toxins are good examples of how nature has evolved into getting this defense mechanism against predators. Now, what's interesting is that tamoxifen does not act like the other two toxins. And instead, what it does is it targets inactivation and it lengthens the time it takes for the channel to recover from the inactivated state. So I'm going to prove to you later how I found that that discovery. So the uniqueness led us to explore the possibility that tamoxifen might be used as a therapeutic. Well, in fact, tamoxifen is already a therapeutic if you heard of it. It's an estrogen receptor antagonist, and it's given to patients with breast cancer. With breast cancer. So why are we testing it? It's because we have also found that it has off-target effects on sodium currents. So we want to see whether this, or like how it's affecting the sodium current. So upon administration, tamoxifen in the body is going to be metabolized to the liver into either 4-OH tamoxifen, into NDAS tamoxifen, and also in endoxifen. And these metabolites are going to circulate around the body in different abundance. So what's interesting is this one right here, NDAS tamoxifen. So even though it has the highest abundance in the blood, it actually has low affinity for estrogen receptor. So that means there is some sort of receptor specificity where NDAS does not like the estrogen receptor. And we want to see whether we can use this to our advantage using NDAS tamoxifen as a template and somehow develop a small molecule where this ligand can target just sodium currents and not other channels or other receptors. But of course, first we would need to make sure that these metabolites actually have antagonistic effect on our sodium current and how potent they are as well. So the method to doing all that, to measuring potency, is to use whole cell patch clamping. So here on the left is a typical electrophysiology rig. And because we're working with hex cells are usually just 10 to 15 microns. And therefore, we need to do everything under the microscope. So the day before an experiment, I would transfect the cells with DNA of the sodium channel. So the reason is overnight, the cells are going to express the sodium channels and they're going to travel to the cell membrane where I can do patch clamp on them. Okay, so briefly, patch clamping is just forming a strong seal between the small glass electrode and the cell that's overexpressing the sodium channel. So how we would do it is, this is our small electrode up there. So we're going to approach the cell and then have it right on top of the cell. And because the membrane is fluid, so it's going to just attach to the electrode and try to form a seal on it. And once the seal is achieved, I'm going to break into the cell so that I can have access to all the channels that are on the membrane at that point. Now with that electrode, I can also manipulate and also mimic the stimulus that's given to a cell. So I could like depolarize the cell and tell the sodium channels to open. And then I can measure the resulting current as well. So here I have a simple protocol where I start off with the closed state, and then I give it a depolarizing pulse at -20. So at this point, the channels should go from closed to open. And like I told you earlier, on its own, the sodium channels are going to inactivate So here you're seeing the inactivation curve, even though I'm not changing the protocol at all. And then at the end, I'm just going to reset everything and force the channels to close again by voltage. And from here, I can keep running this protocol over and over again, but then I'm going to exchange the bath solution. So the cell is going to experience different environment. So what I'm going to change the bath solution to is just different and increasing concentration of tamoxifen or its metabolites. So here we have in black the control, and then light blue is just a small amount and an increasing amount until we get to the deeper blue. And you can see that the current is reduced with increasing drug dose, and that's expected for an antagonist anyway. So we can plot the percent inhibition over the drug concentration. And this graph here would give us an IC50. So we can look at the IC50, and that gives us basically a measurement of the potency. And we found that among all the metabolites here, all of them have pretty much the same potency in the sub-micromolar range. So we started doing bacterial channels just because our collaborator had the crystal structure in the bacterial version. But we also want to know whether this all these effects are translatable to the human version as well. So that's why I also performed the same experiment, but on the human NAV 1.2 subtype. And here we found that, again, all the metabolites have similar inhibitory effects, but then it's a little bit less potent than the bacterial version. Okay, and next, in collaboration with Bonnie Wallace from the University of London, we solved the structure of the bacterial channel in complex with all these metabolites. And just like the potency experiments, all the metabolite structures actually look the same. So I'm just gonna, I guess, simply call them the map bound structure. And so here in gray is the unbound structure, and in red is just our new bound structure. So you can see that overall, everything looks kind of the same, and the only big difference is over here in the at the bottom. And so that's our S6, which is displaced slightly towards the pore, causing a constriction in the exit of the conducting pathway. And tamoxifen, interestingly, actually binds to 2 sites per subunit, making it a total of 8 drug molecules in the complete channel. So here we label them the outer site and the inner site. And here in orange is the outer site. And the outer site, we think it binds with weaker affinity, just basically based on the crystal lattice parameter, because they look more disordered. Whereas the green one here, the inner site, seems to be more defined when we were solving for the crystal structure, and also is more buried in the middle. When I quickly looked at the interactions, I saw that there were some hydrogen bonds. So then I tried to mutate those bonds, and also noticed that the potency was decreased, telling us that this inner site is probably important, and it's probably involved in tamoxifen binding. And since this tamoxifen is near the inactivation gate, we think that what we're seeing right now is a non-conducting state that is somehow related to inactivation. And so the next bit is just going to be about solving the mechanism of how tamoxifen affects the channel. So we think it's inactivation, and we looked at the onset of inactivation and that between the drug and the gas control, and that didn't change all that much. So therefore, we looked at the recovery of inactivation. So we call that back then we had, after resetting the channel from inactivated to closed, we waited long enough so that all the channels were back to closed state. So I modified the protocol here, where I'm going to run the same activation pulse, but then back to back. And I'm going to vary the onset of the second wave. So you can imagine if the duration is too short, there's not enough time for the channels to recover. So there's going to be a population of channels that are still unavailable for the second for the second wave. So then the second current is going to be small, just like right here. Okay. And then on the other hand, if there's an extended recovery time, then there should be enough time for the channels to recover. And then we should have a much bigger current. And so here we have in black the control, and here in blue the 4-hydroxytamoxifen. And we notice that the time it takes for the channels to recover is actually 10 times longer for for 4-hydroxytamoxifen. And therefore, we propose that tamoxifen and metabolites act by slowing the recovery from inactivation. So you might not be convinced yet, and neither were we. So therefore, we went back to our simple protocol, but this time we want to test the state dependence of binding and also on the inhibitory effect. So the difference this time is that we're going to stop depolarization while we apply the drug for a minute, and then we wash off the drug, so that's back to the control condition. And all of this were done in the absence of depolarizing pulse. So the channels were closed this entire time. And after that, we start to depolarize the cells again to observe something interesting. So the first pulse right after the drug application didn't seem too affected, but then the second one is very affected. And so there we were trying to explain why that would happen. So it appears that the drug can still bind to the probably the binding site, or it can still linger around, but it just has no effect while the channels were not activated. But then the drug finally could take effect after the first set of channels were activated and inactivated. And then they got temporarily trapped in the inactivated state just because of the slowed recovery. So this leads to the lowering of the available channels, and therefore the second pulse would have of the drug depends on into the inactivated state. And finally, we demonstrated further by shortening the depolarizing time. So instead of allowing 150 milliseconds for the channels to fully go from open to inactivated, we shortened it to only 7 milliseconds to minimize inactivation. So basically, we're just bouncing between open states and closed states and not letting the channel to have time to enter the inactivated state. So doing the same experiment again, we can see that we're not losing current nearly as quickly. And again, confirming that efficacy requires going into inactivation first. And it's also driven by the impairing of the recovery of the stabilization, and it stabilizes the inactivated state. So based on this mechanism, this is what we think the drug is doing in the neurons that can, and what it can do to a neuron that's firing aberrantly after repolarization. So we're hoping that tamoxifen is going to bind after the first action potential, and it's going to block any extra sodium channels that are firing. And in that sense, we can return the action potential back to normal. So after publishing these results, we transferred this hypothesis from the bacterial channels to human and to in vivo pharmacology. And so here we are still working on the manuscript. So I can't say too much about the second paper, but then this paper was published in Molecular Cell, I think earlier last year. So you could look for it if you want to read it again. And so this gives us a first glimpse that maybe this receptor site can help treat epilepsy or neuropathic pain. Okay, and then the second project is my PhD project. And so it's not a nerve cell, but then it's still it's a cardiac cell channel, which is also excitable cells. So in order for them to communicate, it's still through action potential. And so then each time, so like we can I guess breathe and think about a heart rate for a second. So So each time the action potential fires, the heart is going to beat once. And I'm going to probably assume that my heart rate is probably slightly faster than yours right now, just because I'm a little nervous. So adrenaline also increases the firing of action potential and therefore increases heart rate. And both of these properties is because of the pacemaker channel, or abbreviated HCN, hyperpolarization activated cyclic nucleotide gated channels. So here we have adrenaline that increases the firing of action potential. And so HCN is very much similar to the bacterial sodium channels. Again, they have 4 subunits, and they assemble together in a tetramer around the central pore. And it also has the VSD, S1 to S4, and it also has the pore domain, S5 to S6. So sometimes the HCN channels is called the funny channel, but not because it's like haha funny, but it's because it has like an unusual activation property. So instead of like sodium channels, which activate during depolarization, HCN channels activate during hyperpolarization or just very negative voltages. So this is actually pretty good physiologically, because that means that channels can be opened during resting membrane potential. And the influx of sodium channels then can trigger the activation of sodium channels, which then can trigger action potential firing. Another odd feature about HCN is that it is also modulated by cyclic nucleotide. And that's because of a domain here in the C-terminus, which is also what I'm going to be mostly discussing today. So the C-terminus contains the CMBD, or cyclic nucleotide binding domain, and a C-linker, which links the CMBD to the rest of the channel. And during fight or flight response, adrenaline is released. So that activates G-protein coupled receptor, which then increases the production of cyclic AMP. And the direct binding of the secondary messenger to the CMBD is what modulates and increases channel opening, and increases the action potential, and increases the heart rate. And here we're looking at the response of cyclic AMP to HCN channels. And up here is in the absence of cyclic AMP, and in the bottom is in the presence of saturating amount of cyclic AMP. So it might not make a lot of sense because they're current traces, and but like this might make a little bit more sense in this graph here. We have the fraction of currents that are activated versus the range of memory potentials. So here in the open square is without cyclic AMP. And so that means that the channels are only active during this range. However, when cyclic AMP is added, it increases the open probability. So channels are more active in a larger range, resulting in this signature rightward shift that we're going to see again later. So because we don't have too much time, so I'm not going to go into too much detail on this project, but I'm just going to show you one of the pieces of data. And so just like the other project, the CMBD seems like an appealing therapeutic binding site, right? So we could maybe produce a drug there that could, I guess, alter the binding of cyclic nucleotide. So cyclic nucleotides, there are a lot out there, and I also tested quite a few of them back then. But then today I'm just going to show you the one that's most interesting, at least the one that I think is the most interesting, and that's cyclic CMP. So on the left is with cyclic AMP. So AMP has the adenine purine ring up there, whereas cyclic CMP has a pyrimidine ring up there. And when you look at the activation curve that we saw earlier, again, cyclic AMP has the signature rightward shift, whereas cyclic CMP has a way smaller shift, and it's about like 30% in magnitude. So we will classify this not as a full agonist, but only as like a partial agonist because the shift is way smaller. And so here in this talk, I'm going to talk about a few experiments that I've done to compare cyclic AMP, a full agonist, with cyclic CMP, a partial agonist. The first the first experiment was called DLS, because we know that the C-terminus would tetramerize normally in order to form a channel. So DLS would tell us the molecular weight. So how it works is DLS shoots a laser at the solution with purified protein, and the different size of the globular protein would have different Brownian motions, and therefore it would scatter light in a different pattern. And using that scattering pattern, we can look at, we can calculate the apparent molecular weight of the protein. So here I have plotted the apparent molecular weight. The molecular weight of a monomer is about 25, whereas for a tetramer is about 100. So when you see a number in between those, it's probably just a mixed population of tetramer and monomers. And on the x-axis, we have increasing protein concentration. So now let's look at the no ligand curve first. We see that even on its own, even without any ligands, the tetramerization would increase just with protein concentration. And that's probably just due to law of mass action, where when you have more concentration of something, they would just interact with each other. And then when we look at cyclic EMP and GMP, both are full agonists. You can see that at non-tetrameric concentration, for example, at 100 micromolar of proteins, you see an increased population of tetramers, telling us that a full agonist would promote the tetramerization of the proteins. Now, what about the partial agonist? So here in blue is our cyclic CMP, and you can see that under any concentration of protein, cyclic CMP has no additional effect on any of them. So you might be wondering, oh, is this like a pyrimidine thing? Like a pyrimidine probably just doesn't have any effect. So I have a control here. Cyclic UMP is also a pyrimidine. And here, cyclic UMP is a full agonist, and it's able to tetramerize our protein. So it's not a pyrimidine. And then another thing we might be thinking is maybe cyclic CMP just doesn't bind to the CMBD at all. And therefore, the next test was the ITC, the isothermal titration calorimetry, which would tell us the binding affinity of ligand to proteins. So here in a very well-insulated cell machine, we have a reference cell, which is water, and the sample cell is our purified proteins. And here I'm using 200 micromolar of the protein because that's where the protein is predominantly a tetramer already. And then when the experiment starts, I'm going to fill the sample cell with just injections of the cyclic nucleotides. And so one microliters at a time, the machine is going to detect the heat change that occurs upon the interactions of ligand with protein and spit out a isotherm like this. So on the top is the raw data for the heat. So the y-axis is the heat and the x-axis is the time. So each downward inflection point is basically the amount of heat that is released when cyclic EMP reacts with or binds with the protein. And you can see that as the reaction progresses, the amount of heat decreases and it goes back to baseline, right? And that's just because all these binding sites have been saturated. Now, the amount of heat at each inflection point can be integrated to generate this isotherm down here. So the binding affinity is basically the slope of the isotherm. But then, so the problem here is when we look at it, we can see that there are two slopes, and that's because there are two different binding events. So we fit this curve to a model, a two independent site binding model, and so then program will tell us the stoichiometry, the binding affinity, as well as some thermodynamic parameters. So here we see that there are two binding events. The first one is here on the left, and that one binds with 0.13 micromolar, whereas the second binding event over here binds with 1.83 micromolar. And the stoichiometry is telling us it's about like a 25%, 75% distribution. So that tells us that maybe 1 out of the 4 subunits is binding with the high affinity, whereas the other 3 is binding with the lower affinity. And we call this term negative cooperativity, because after the first binding event, there's probably some conformational changes that makes the binding harder, or like makes the binding site different and with lower affinity. So then the other 3 would have lower affinity binding site. So for cyclic CMP, according to the ITC, we only see one binding event, and the stoichiometry tells us it's 1. So it's not the 25-75% distribution like we see in the other one. And also the affinity is lower. So this tells us that cyclic CMP does in fact bind, but then it binds differently. I also had the crystal structure for cyclic CMP, but then since it binds to the same binding site, I'm just not gonna show you. But then crystallization was also another evidence showing that cyclic CMP does in fact bind, and it binds similarly to cyclic AMP. And in summary, so like I told you earlier, I've tested a lot of different cyclic nucleotides. So these guys are just all kind of part of the same group. I co-crystallized all of them to the protein, and all of them bind to the same binding pocket. And among that list, only 2 of them were partial agonists. So everyone had the significant shift except for cyclic CMP and cyclic IMP. And what's also interesting is those 2 partial agonists never promote tetramer, like you've seen earlier, and also they're the only ones who produced the single site. So that might make sense because if the tetramers are not being formed, they might be missing some communication, and therefore they're not able to execute negative cooperativity. So this information here is physiologically relevant because non-canonical cyclic nucleotides like cyclic C and cyclic IMP, they also exist and they also synthesize in the body. So looking at the KD values that are measured, they are also in the physiological range. So they might have some action in the cell. And the fact that they're available in the cell might suggest that HCM regulation might be more than just an on and off, but rather it might be like a spectrum of activation in a sophisticated manner, which involves just different cyclic nucleotides at different time. And even though cyclic CMP and cyclic IMP, we call it partial agonists, we can also call them partial inhibitors because they are reducing the effect of cyclic AMP, and they can outcompete, they can compete for the same binding site as cyclic AMP. So that might be interesting. And it's just like the pharmacology of N-tamoxifen earlier. We could make the partial agonist with a stronger affinity, and then the drug can then outcompete for the binding site. And hopefully that could be a drug that can be used to control cardiac arrhythmia. And with that, I'd like to thank all of you for listening. And I'll also like to thank Paul and Eric for their great mentorship over the years, and also the 2 labs for their continuous support and friendship, even though we're not working in the same lab anymore. Right? And with that, I guess I will take any questions you guys might have.
Thank you very much for your talk. Do you have any question from the audience? You can use the Q&A to type your question, or you can raise your hand and we'll give you the. So we have 2 participants that raise their hands. So Mark Rio, I will give you. Mark, I think you can ask your question.
Can you hear me there?
Yes.
Oh, wonderful. Thank you very much. So Leo, wonderful talk. Very interesting. I really enjoyed that. I have a very fundamental question. When I look at your tamoxifen data, you did this on bacterial sodium channel, voltage-gated sodium channels,
Yeah.
which are kind of that tetramer and different from humans in that they don't have that cytosolic linker regions between the transmembrane domains.
Yeah.
So the classic model of voltage-gated sodium inactivation is that it's the 3 to 4 transmembrane domain cytosolic region that's the inactivation gate. If that's absent, how do those channels inactivate?
Right. So right now we don't have a model for inactivation yet, and we also don't have a, I guess, structure of an inactivated model. So all we can say is that the bacterial channels do inactivate, but then we don't know which part of it is involved yet.
So like this, in your one figure, you showed tamoxifen binding to the bacterial model that you had, but you didn't show it binding to the human model.
Yes. So right. Okay. So the reason is because we published the structure in bacteria, but then we didn't do it in human.
Okay.
So that's why we don't know where it binds yet in human.
Yeah. So that it looks like probably in bacterial, like we know that bacterial inactivation is always, the rate is slower than it is in human voltage-gated sodium channels. So do you think that there's actually a completely separate inactivation mechanism in the bacterial tetramer? Like it's something in the S6 domain or something like that is doing that?
Yeah, so it's definitely possible, and I would not be surprised if it's different. Yes.
Okay, perfect, wonderful. Thank you very much.
Thank you.
We have another question from Zoe Zun.
Yes, thank you so much. That was such a fantastic talk. I really appreciate it. And I think Mark actually asked a couple of questions that I had already. I guess I was wondering too about how tamoxifen bound to human sodium channels, but I'm not sure if you know which sodium channels tamoxifen can bind. Can it bind to sodium channels in various different cell types, neurons, cardiomyocytes, skeletal muscle cells? Like, are you able to test that in cell culturing? And if so, what would happen, do you think?
Yeah. Yeah, so that's the data for the next paper. So that's that's something like I'm not sure if I can talk about yet, but so we can certainly test it using the same I guess protocols. So instead of transfecting with the bacterial version, all we have to do is transfect it with different subtypes of the sodium channels. So in sodium channels or in humans, there are 9 subtypes. So NaV 1.1 to 1.9. So those, like the cardiac one, for example, is in NaV 1.5, or like the skeletal one is 1.4, and the neuronal ones are like 1.1 to 1.3. So yes, we can definitely just isolate different subtypes and test them differently and see whether tamoxifen has an effect on of. So that is done. I just can't say how or what binds it.
Oh, that's really nice. That's really exciting. And I guess the last question I had was, do you know what the half-life of tamoxifen is?
Oh, so that is certainly published. I don't remember it now, but I had it at some point. And yeah, sorry, I don't remember. But it's not that short though.
Fantastic. Well, best of luck with all the rest of your studies. It's really interesting, and I think it's going to be exciting.
Thank you.
I have a question.
Yeah.
So many patients that are under treatment with tamoxifen for breast cancer, for instance, they report brain fog or chemo brain. In light of your results, do you have any idea or hypothesis about what could happen there?
So whether it's like blocking some sort of neuronal signals, maybe. So I mean, it could. I don't want to say that is the cause because we haven't tested it yet. But knowing that it has a side effects on sodium channels, it could totally affect some sort of neuronal pathway.
And I was wondering something, a different line of question here, but so tamoxifen was designed to bind the estrogen receptor, right?
Mm-hmm.
Have you tried estrogen on the sodium channels? Is that doing anything?
Oh, whether estrogen binds to it. Yes. So estrogen does have an effect on sodium channels. Yes.
Oh, okay.
Yeah.
On the bacterial one?
It's not as potent though. So it's like way weaker potency. So the primary target for estrogen would still be estrogen receptors.
Yeah.
So like yeah. So it's not nowhere near the potency of estrogen receptors.
Okay. Yeah. Now looking forward for those new data on the maybe the human form.
Me too.
And
Me too.
have you tried on a primary cell culture, maybe on neurons or on cardiac cells that express both receptors to see if actually tamoxifen has still an effect on the cell that actually express the ERT receptor?
Yeah, very good question. So yes, so I didn't do it myself, but so Joe here was working on primary culture. So she, I believe, was working on DRG culture or hippocampal neurons, and she was testing the action potentials and see whether tamoxifen affects action potentials of those. Yeah, so it'd be like in vivo pharmacology that way.
Okay. Thank you. Yeah. Do we have other questions from the audience? Robin, did you raise your hand? I'm sorry.
Sure. I wanted to ask, I want to completely switch gears and head a little bit more towards the non-science side of this talk.
Yeah.
As someone who graduated relatively recently from UBC and who is now advancing their career in other ways, I know we have a number of students in the audience. I was wondering if you could speak to sort of the challenges or the wonders of that particular journey and how that went for you. I don't know exactly how to frame it.
Wow. Yeah. Yes. So that's certainly a very good question. Let me try and organize my thoughts, because you're kind of like putting me back into like, you know, the depressing time way back then. So no, I'm not saying that PhD is bad, but like if you talk to a lot of PhD students, they will probably tell you that that's one of the toughest time of their life. And it's not so much like the work is hard. It's more just like having the mentality to go through it. So it's like you know you're gonna graduate, but it's just like you don't know how long it'll take and whether like you have the energy to I guess finish it, right? So I think I didn't think I had it too hard because my professor was really encouraging. So he was always there to support me. So like even like when I'm working late hours, he would tell me not to work as hard. So I know like I think some professors are different, where they want their students to work 24/7. But then my professor wanted me to have a better work-life balance. So that was really nice. And that made me, I guess, less stressed. And then the other thing that was really helpful was just the other PhD students that were around you, because you are always in I guess, so like, I guess not in my lab, but like neighboring labs, they all have grad students, right? So it's good to have a good relationship with them and just like talk to them, because you might need the support, but then at the same time, they need the support as well. So that mutual support really helps us get through it together.
Great. What about?
So I'm not trying to discourage anyone from doing a PhD.
Actually, I feel like you're echoing my own experience with PhD, so I think it's true all the way along. What about making the transition from being a PhD to your postdoc? Sort of what came next? How did you make that decision?
So I actually didn't think I was going to do a postdoc.
Mm-hmm.
It sort of just like naturally happened. I don't know how to explain it, but like I just felt like I was just going with the flow, and then it happened, so I just let it happen. So what happened was towards the end of I think it was like a year before my PhD ended. So I went to a conference, international conference with my professor. So Eric was trying to introduce me to different friends and wanted me to do a postdoc. And I kept telling him that, no, I'm not going to do a postdoc. I don't want to be a professor. So that happened. And then when I got there, I was just delivering a talk and a poster. And that was where I met Paul, which was my mentor for my postdoc. So he offered me a job there. And I was like, but I'm not sure if I want to do a postdoc. And I wasn't done either, because I still had like a year to go. I didn't write my thesis yet. But like I kept that in mind. And at some point, like I think I just changed my mind. I thought maybe doing a postdoc is not a bad idea. It's not like I'm going to go into academia for sure. It's just like opening a different door, right? So then I took the offer, and so I just started my postdoc immediately after I graduated.
Awesome. Great. Thanks.
Do we have other questions from the panel or from the attendees? I see Shirley. Yeah, go ahead.
I just wonder, how did you decide whether you want to go to industry or academia, whether you want to do research or teaching? Like, were there any like things influence you, or what kind of character do you think that a person that will be more suitable for which type of career?
I feel like I haven't met enough people to be able to say that, but so I know I'm not the academia type, or like as in like a research professor type, because all the research professors that I've met are really good at selling themselves and selling their projects. I'm not saying that's like a bad thing, but like they're really good at marketing and like really like passionate with what they're doing. And I feel like I always have this doubt, like I don't know if I'm like the right person. So with that doubt, I already know that I'm not the best like marketing person. And at the same time, the thing that worried me about the most about being a research professor is finding my own fundings. So I don't want to have the responsibility to run the lab and also pay for all the, I guess, people under me. So yeah, so with that, like since the very beginning, I already knew that I couldn't be an academia professor. But then I kind of like research and teaching at the same time. So that's why I'm kind of still in the crossroad of like whether I want to just go teaching or just do research. So if I just do research, I'll probably do industry. Yeah. So like right now I'm still thinking. Yeah. Well, yeah. So the character of being a professor, then obviously you have to be able to teach. Because I'm not saying like, I know there are professors right here. So I'm not saying that you guys are one of those. But then you know that there are some professors who are like just not very good at teaching. So those professors should probably not teach. Yeah.
No, we have a great comment, I think, and I really share that comment. Thank you for your honesty on those comments. I think it's really appreciated for the students. There is not only academia after research. I think that's important for everybody to understand that there's a lot of big world outside of university. So yeah.
Yes, and also the pay is better in industry. So if you're more like money oriented, then you should probably not consider academia.
Great. Do we have other questions from the attendees or from the panel? I see one in Mark. Yeah, Mark, you can talk. Yes.
So I'm going to echo those comments. I love this kind of discussion. I think it's fantastic when people are honest about this kind of thing and show students there's many options. So in that line of thought, and also because this is a diversity seminar, do you find there's any, as a person of ethnicity in looking for jobs, do you find there's a difference looking for jobs in industry versus looking for jobs in academia? You know, have there been any roadblocks for you, or do you find there's less roadblocks in one path than the other?
Okay, so I'm not sure if I can answer that yet because I haven't really searched that many jobs. But so I think like the only roadblock so far is just with the visa. So like as a Canadian, getting a job in the US would require a work visa. So I think that's the biggest roadblock I have so far. So it has nothing to do with ethnicity. It's just that some people don't want to sponsor a visa. So that's just an extra thing to consider whether you want to work in the States or stay in Canada.
Great, thank you.
Okay. Any final questions, maybe? I see none. Okay, so we can all thank you very much, Dr.
Thank you.
Leo Ng. Thank you very
Thank
much for your talk and for your answers to all those questions and your honesty about academia versus industry and so on. That was really, really, really great. Thank you very much.
Thank you. Right, bye.